So to be a science, science has experimentation to verify our hypotheses. Once you're ready to share your results, they need to be peer-reviewed. And we see catastrophes happen all the time when scientists decide to go straight to press conference without allowing their work to go through peer review, because sometimes they're completely wrong about what they think they found. Experiments must have control groups, and that's what we call a controlled trial or a controlled experiment. That means there's a group that does not get the treatment. And we need control groups, whether you're talking about chemistry, cells in a dish, or you're testing new medicines. Results have to be repeatable by anybody, and if your scientific results cannot be replicated by other labs, then something is wrong. Either you were sloppy or you're manipulating your data or something. Luckily, that's rare. Science only deals with the natural world. It is not the only way to know about the world. It's not the only type of knowledge. But it's the best way we know of to figure out how things in the world work. Results have to be objective, meaning that anybody could see that same result. Not a matter of opinion, not subjective. Let's talk about a hypothesis versus a theory. Hypothesis is a preliminary idea. It's an idea of how things work. You might have data for a hypothesis. You might not have any data at all. You might just be doing a thought experiment. Hypotheses change a lot. We cannot get too attached to a hypothesis because data could mean that we have to throw it away. A theory, on the other hand, in science, if we call something a theory, it is backed up by years of data and experiments. It is generally accepted by the scientific community as being a good explanation for the data that we see. Theory in everyday speech means a preliminary idea. So if you say, well, I lost my car keys, but I have a theory about where I left them, that is a hypothesis, not a theory. So theories, any scientific theory could be thrown away with new data. No scientific theory is above criticism. And sometimes theories that have been around for hundreds of years do get overturned, and I will show you some examples of that. They don't get overturned very often, though, because to even become a theory, it has to have a lot of data to back it up. Science is always changing, and this is sometimes disconcerting for the general public when they see science happening in front of them, like over the course of the pandemic. Scientists change their minds and have new hypotheses as new data comes in, and people say, well, they don't know what they're talking about then. They keep changing their minds. Yes, that is exactly how science works. Our conclusions are based on the most current data. So if there was a paper published 2 years ago that showed something interesting, and since then 10 more studies have been done that disprove that and showed that that was a bunch of garbage, we have to go with the most recent data, and our hypothesis, even though it was interesting, has to go in the garbage can. So some of the big theories that we're going to talk about over the course of the semester. One of the first ones that we're going to talk about in the second module, we're going to talk about cell theory. Cell theory replaced an earlier theory called the theory of spontaneous generation. Cell theory has 3 important parts. One is that all cells come from other cells. Right now on Earth, there aren't any new cells appearing from non-cells. Although we'll talk about the origin of life at some point. That had to happen. But right now, every cell on Earth is from the division of another cell. Cells are the smallest thing we consider alive, so if you take a cell apart and take out its constituent parts, like we take out the DNA or take out the chromosomes, we no longer consider those bits to be living. Every cell has a membrane, and the membrane is the outer border of that cell. The person who first provided laboratory experimental data for cell theory was Louis Pasteur. You've probably all heard of Pasteur. His experiments replaced the previous theory called the theory of spontaneous generation, which was around for 2,000 years at least. Probably we could blame that one on Aristotle as well. And the idea of spontaneous generation was that life is spontaneously appearing from non-living things. People could see evidence with their own eyes. If you have freshly cooked soup, for example, and you take a drop and put it under the microscope, there's nothing alive in there. If you leave it out for a few days and you look at a drop under the microscope, oh, now it's full of cells. There's bacteria in there. Eventually there's going to be fungi growing in there. If you leave it out long enough, you're going to have worms and maggots and flies hatching out of that soup. This seemed to be great evidence to people that life was evolving in that soup, and simpler life was turning into more complex life, and those first cells that appeared were just appearing spontaneously. There was even a recipe for making mice that appeared in the Farmer's Almanac in the early U.S. publication, which was, what was his name? Benjamin Franklin, his little publication. So to make mice, they said if you took a dirty work shirt, so sweaty, dirty work shirt, put it in a burlap sack with some wheat grains and close it up. If you came back after, and then throw it in the corner of your barn or something, if you came back after 21 days, those little microbes will have turned into mice. It sounds ridiculous now, but I mean, this was considered a theory that had evidence. So Pasteur said, I don't think that's true. Pasteur was working with bacteria in his lab, and he kept getting the same species of bacteria over and over and over. And he said, well, if they're just appearing spontaneously, why are they always the same? Why do I keep getting the same species? And so he came up with a new hypothesis, and that was that there are bacteria in the air and fungal spores in the air, and they are just landing in the growth media and growing. So nothing is appearing spontaneously. In fact, they are growing from other cells. And so his experiment to test that, he invented this little flask and had somebody make them for him, and they were open to the atmosphere. So he used beef broth for most of his experiments, clear broth, because it's very easy to tell, as you'll find out when you're in microbiology. If your broth gets contaminated and starts growing bacteria, it becomes cloudy. So if you're looking at that broth and it's still clear, there's nothing growing in it. And they knew that if you boiled liquids, you would kill all the organisms in there. So this particular type of flask with this spout, nothing could blow in there, but it still allowed the exchange of gases. So oxygen and carbon dioxide could exchange. And so for his control, he took the same flask, boiled the broth to sterilize it, and then broke the top off. So it had access to things falling down in the room. And that one, of course, became contaminated and grew bacteria. The one that was open to the atmosphere, but nothing could fall in there, no growth. There's a museum in Paris where they still have some of his flasks from the 1860s. Of course, it's, 150-year-old beef broth. But you can still see that the broth is clear and that there's nothing growing in it. So this proved that it was not spontaneous generation that was leading to microbial growth. In fact, these microbes were already in the atmosphere and were just blowing into and contaminating whatever it was. And you'll find this out too if you're going to take micro, that you have to just think as you do things that there is just bacteria and fungal spores just raining down on you constantly. And even when you open your Petri dishes, you just have to just like quickly open them or open them upside down, because otherwise it's going to have everything in there. So that was the first experimental evidence for what we now call cell theory. We're also going to spend a lot of time talking about the theory of heredity. And the theory of heredity is both about DNA and the structure of DNA and how those genes are expressed. And we're going to talk about genetics and Mendel, the first person who kind of started the field of genetics. And we'll also talk about this, and that'll be Module 4. And we'll talk about the structure of DNA and the discovery of DNA and the background on that in Module 5. We'll talk about energy conservation. Energy from the sun is the fuel for all life on Earth, one way or another, through photosynthesis. And we will talk in Module 3 about photosynthesis, about respiration, and how energy isn't created, it is converted from one form to another. So all the energy for life on Earth is ultimately coming from the sun. Every bit of energy that you are using right now came from photosynthesis. All the energy in the ATP that's being used in your body came from the sun, and that energy being converted by photosynthesis into energy in some plant which you ate, or you ate an animal that ate that plant. That's the conservation of energy. We will talk about evolution, and we'll also talk a bit about the origin of life, which is kind of the beginning of evolution. We have several labs that also have to do with evolution. So here's our 3 domains here: bacteria, archaea, and eukaryotes, and our very ancient split. So at the beginning of Module 2, we're going to talk about the origin of life down here, and this ancient split and the origin of eukaryotes a little bit as well. It's hard to talk about anything in biology without touching on evolution in some way, because living things have evolved and are continuing to evolve. Everything that we consider alive today has a common ancestor that first appeared about 3.8 billion years ago, which we'll talk about. There's 2 basic types of evidence for evolution. One is the fossil record, ancient organisms, things that are extinct for the most part, and comparing them to things that are living now. The other big piece of evidence for evolution is looking at things that are alive today. And we're all very familiar with the fossil kind of evidence. Everybody's heard about dinosaurs and other extinct things. Here's another extinct organism, the Basilosaurus, which is, from the name, you might think it's some kind of dinosaur, but it was misnamed. It was lived long after the dinosaurs went extinct. It is, in fact, an ancient whale. So the name sticks, even though the person who discovered it didn't correctly identify it as an ancient whale. So we can compare fossil organisms to organisms that live today. And we'll talk more about whale evolution during our evolution chapter. But we can also look at things that are alive today and comparisons. So this is a nice SEM image of a paramecium. And we're going to hopefully look at live paramecium in lab. Paramecium are single-celled protists, and they are covered with these fine hairs called cilia, and that is how they move. Cilia can move, and we'll also talk about that in the next module. We also have cilia. We have cilia in several places in our bodies, but especially inside the bronchioles. They move the mucus and all the crud that sticks to the mucus, they move it out. And they move just like the cilia on a paramecium move. And there's more similarities than that. If we use an electron microscope and slice through the cilia of a paramecium and a human, in fact, we find the exact same structure. And it's made up of these little proteins called tubulins that are arranged into these long straw-like structures called microtubules. And we now can look at the genes. And Paramecium have genes for the tubulin protein. We have genes for the tubulin protein that are very similar. So the best explanation for this, why do humans and these single-celled organisms have this almost identical structure that works the same way, is made up of the same proteins, and in both cases it moves fluid. That's what cilia do. And Paramecium is swimming through the fluid. In our case, it's pushing the fluid out of our lungs. The best explanation is that we evolved from a common ancestor who had cilia. So this is a summary of evolution, and we will talk about evolution in more detail in Module 3. But the basics of evolution, if you've never heard it before, individuals in a population vary. They are different from each other. This variation can be passed on to the offspring. It's heritable. All populations produce more offspring than are going to survive, some spectacularly so. Like for houseflies, only about 1% of those houseflies are going to survive to adulthood. Individuals that are better adapted to the environment are much more likely to survive and reproduce, and over time, this difference in survival is going to change the population. So individuals don't evolve. Species and populations evolve and change generation after generation. Another phrase we hear along with evolution is survival of the fittest. So in evolutionary terms, fitness means surviving to reproduce. That's it. Fitness in evolutionary terms doesn't have anything to do with stronger, faster. It's whatever will help an organism survive in that particular environment, and it's very environment specific. So a fitness in a particular environment might be very unfit in another environment. Like a cactus here is a plant that is supremely adapted to live in very hot, dry environments with very low water availability. They have no leaves. That helps them conserve water. They're just a big fat stem. And to protect themselves from larger animals taking their water, because of course they're full of water, they are covered with thorns. So in a desert, this is a really good adaptation, all of these things. But if you took that cactus and planted it in your yard here, it probably wouldn't even last the summer because it's too wet, it would rot, it wouldn't survive the winter. It's just not adapted to our environment. Although we do have a native cactus in Illinois, but it doesn't look quite like that. But we do have a native cactus. If you would like to have cactus outside in your yard, there is one that lives around here. It's very slow growing though, so we don't see it a lot. So fitness, it has to do with the environment. So if the environment changes too much, an organism may no longer be adapted for that environment. This is one of my favorite evolution quotes from an early 20th century evolutionary biologist, Theodosius Dobzhansky, who is a Russian scientist. Nothing in biology makes sense except in the light of evolution. And this is even more true today than when he first said this. Also, my other favorite evolution saying, which has been put about in various forms since Darwin, is that evolution, it's not stupid if it works, because mutations are random, and what is the best in a particular environment, or what might be the best, isn't always what evolves. And these are a couple of my favorite really crazy evolutionary experiments. This is a recently discovered group of spiders. They're very tiny. They only live in Australia. There are a dozen or so different species, and they're called the peacock jumping spiders. And the males display, and this is the most colorful one. There are others that aren't quite as colorful, but they do these. They're tiny, tiny little spiders, and they do these great little dances. You can find some really good YouTube videos made by the scientists who study these. And all kinds of crazy stuff can evolve. Sea dragons are a type of seahorse. You've probably heard of seahorses. They are very, very camouflaged, but they're just regular bony fish. They are related to tuna and goldfish. They're just fish, but they have undergone extreme adaptation for camouflage at the expense of being able to swim fast. So they're super slow swimmers, and their big claim to fame is camouflage. So we're going to talk a lot more about evolution in Module 3. So to wrap up this chapter, science starts with observations. We want to know how something works. So we form a hypothesis and then test that hypothesis. And you can test your hypothesis either with an experiment or by making more observations. If your hypothesis is not supported, you have to throw it away or modify it. And this is what makes science different from other ways of knowing about the world. Once you're ready to share your ideas with the rest of the scientific community, you go through a peer review process before they can be published. And so things that are just bad science usually get caught at some point. We're going to talk over the course of the semester about the big theories in biology: cell theory, heredity, energy conservation, and evolution. All right, on to chemistry.